Hi, I'm Roxanne Sebastian. Today is February 2nd, 2019, and this video is the part 2 of week 4 lab 1, which is the methyl red test. So on part 1, we prepared our MRVP broth and put it on the incubation area for 48 hours, and then after 48 hours we have to observe for growth, which appears to be a turbidity, and I've already took pictures and put it on our lab exercise. So for part 2, we are asked to sterilize 4 test tubes, and then you need to label 2 test tubes E. coli, just this one, and then 2 test tubes will be labeled S. epidermidis. So once you've labeled all your test tubes, you're going to carefully pour the content of your MRVP broth onto the corresponding test tube. So you're carefully going to pour half of the incubated MRVP broth into the E. coli. So that's half. I've already poured it earlier. And then half and half on the S. epidermidis test tubes. So you should have approximately equal amounts of broth in all 4 test tubes. And then next, you're going to get your methyl red agent. It's you can get it from our kit, from our lab kit. So you're going to get your methyl red agent, and then just you need to cut it in the trash can. Okay, and then you're going to pipette 7 drops of methyl red agent into the E. coli, and the other one 7 drops on the S. epidermidis tube. So this is the E. coli. So 1, 2, 3, 4, 5, 6, 7, and then another one for that's E. coli, and then S. epidermidis. So 1, 2, 3, 4, 5, 6, 7. Okay, and then you're going to observe the color. So for E. coli and S. epidermidis, they're both red, and if the test is positive, the broth will appear red or pink in color. So as you can see, both of them appears red in color. So that's part 2, and then for part 3, the Vogue's Proskauer test, we're gonna need this. We're gonna do the same thing. So for the part 3, we're going to get the Barrett's A agent, and then you're going to pipette 12 drops on each test tube. So you can just cut it. We're going to add 12 drops. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then also you're going to do the S. epidermidis. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Then you're just going to swirl it. Okay. Then after that, you're going to get the Barrett's B, just this one, and then you're going to pipette 4 drops. So 4 drops on each test tube. So 1, 2, 3, 4, and then 1, 2, 3, 4. There's that. Okay, so you're going to shake the tubes for 30 seconds. And then you're going to place the tubes in the test tube holder, and then let them sit for 30 minutes incubation period. And then after 30 minutes, you're going to observe for color changes, and the formation of red on the surface is a positive test. So that concludes my video for lab week 4 lab 1. Thank you.